My name is Caitlin Bowen. I was a graduate student in the lab of Hal Dietz at the Johns Hopkins University School of Medicine. Today, I'll be talking about a story called Endothelin-1 contributes to vascular rupture risk in vascular Ehlers Danlos syndrome mice. So previously, we had performed something called RNA-seq on the thoracic aorta of mice that have a collagen mutation and mice that don't. RNA-seq looks at the genes that are expressed in the aorta, and we can compare mice that have VEDS to mice that don't. When we utilized pathway analysis, which basically analyzes the list of differentially expressed genes, it predicted that there were two upstream regulating pathways that are the major mediators of the observed differences. These were MAP kinase signaling and G protein coupled receptor signaling, or GPCR signaling, specifically through GQ receptors, which culminates in this PLC, PKC, ERK activation. And to demonstrate the relevance of this pathway to the risk of vascular rupture, we found that with MEK inhibition, we observed 94% survival, as shown in this purple line here, compared to only 50% survival in VEDS mice that were treated with a placebo. These are survival curves where the percent survival is on the y-axis and the amount of time is on the x-axis. So in this case, 50% of mice have died by the end of the trial, compared to 90% of mice that are alive on drug treatment. We also observed the same phenomenon with PKC inhibitors, where with 94% survival after 45 days of treatment, compared to only 50% survival in those with no treatment. And while attenuation of this pathway really affords complete protection from vascular rupture in mouse models of VEDS, the mechanism by which this intracellular signaling cascade is activated secondary to mutations in COL3A1 remains unknown. So we thought about different GQ receptors that are expressed in the aorta, and we looked at a number of them. Losartan, an angiotensin receptor inhibitor, had no effect on the risk of vascular rupture. Neither did inhibition of a receptor called thrombin, and neither did inhibition of tyrosine kinase receptors. However, we did find that a molecule that inhibits the endothelin receptor led to 80% survival after 40 days of treatment, shown in purple, compared to only 50% survival in those with no treatment. So what is the endothelin receptor? It's activated by a small molecule called endothelin-1, which is secreted by endothelial cells, or the thin lining of cells. These cells signal through this PLC-PKC ERK axis through both paracrine and autocrine signaling, meaning that they signal to their own cells as well as to other cells, like smooth muscle cells. And since endothelin receptor antagonism was protective in VEDS mice, we hypothesized that we might see more of its ligand, endothelin-1, in the aortas. And as we expected, we saw a lot of endothelin-1 in aortic lysates of predisposed Black 6 VEDS mice. But interestingly, if you watch the other presentation about the 129 mouse model, we saw that that was not different from that observed in control litter mates. And informatively, treatment with the endothelin receptor antagonist, which is called bosentan, did correlate with a reduction in PKC and ERK phosphorylation. We then looked at the activity of endothelin in mice that had been treated with either PKC or MEK inhibitors, which I previously showed lead to profound protection from vascular rupture. Here, endothelin is shown in red, with nuclei shown in blue, and these are aortas from mice. As you can see in the VEDS mice, endothelin appears throughout the thickness of the aortic wall. In Black 6 VEDS mice that have been previously treated with either a PKC inhibitor or a MEK inhibitor, there is reduction in endothelin levels with less red signal showing, with PKC antagonism leading to really profound suppression. So these data led us to believe that there's a positive feedback loop with endothelin and PKC, as PKC inhibition suppressed extra endothelin protein, while endothelin antagonism suppressed PKC activation. So far we've shown that there's increased endothelin in VEDS mice that are susceptible to vascular rupture, and that endothelin receptor antagonism protects VEDS mice from rupture and correlates with reduced PKC activation. There's decreased endothelin in VEDS mice that are protected from vascular rupture, either by pharmacologic intervention, such as treatment with a PKC or MEK inhibitor, or by natural genetic protection. So we then ask the question, what's really the primary cell type in VEDS in vascular dissection? Is it the smooth muscle cells that make up a majority of the aortic wall? Or is it fibroblasts that are the main producers of collagen? Or potentially, is it these endothelial cells that are making and secreting this protein endothelin-1? So given that endothelin-1 was detected throughout the aortic wall in VEDS mice, we asked whether deletion of this gene in either endothelial cells or vascular smooth muscle cells affected survival. So we can actually test the role of endothelin that's made by endothelial cells by deleting the endothelin gene only in endothelial cells. We found that when we did this, we saw improved survival, with 75% of mice surviving to 90 days of age, compared to less than 40% of mice that do have their endothelin gene in their endothelial cells. When we looked at the role of smooth muscle cells by deleting endothelin in smooth muscle cells, we found that it had no effect on survival at all. This led us to want to understand the role of endothelial cells, specifically given that all of our previous analysis was done on bulk tissue. So to do this, we performed something called single-cell transcriptomic analysis. So here we take individual cells and look at individual gene expression. We took over 94,000 aortic cells from 13 different mice, and we clustered them and identified them based on their gene expression. So we found vascular smooth muscle cells, fibroblasts, leukocytes or white blood cells, a small population of Schwann cells, and endothelial cells, shown in green. So we first looked closer at this population of endothelial cells. So we did a gene expression comparison between wild type and VEDS endothelial cells. There are 55 genes that are expressed differently between the two. So like we did with our bulk RNA-seq, we performed pathway analysis. This showed us the same thing: GPCR signaling, MAP kinase signaling, and something called nitric oxide or nitric oxide synthase, which is closely tied to endothelin activity. We're really excited to look more closely at this data, specifically at the different subpopulations of endothelial cells, and how endothelial and vascular smooth muscle cell populations interact with each other in order to learn more about what drives rupture risk in VEDS. So to summarize kind of both of these presentations that I've done, it was previously presumed that a reduced collagen content in VEDS imposes this obligate structural weakness of the tissue. But instead, using both environmental and genetic modifiers of disease risk, we identified an altered molecular signaling mechanism that drives the elevated risk of aortic rupture. We used natural genetic variation to uncover that MAP2K6-mediated p38 activation protects VEDS mice from vascular rupture by inhibition of the signaling cascade. And we uncovered a new role for endothelial cells, and specifically their production of endothelin-1 in VEDS disease pathogenesis, and showed that it's part of this positive feedback loop involving PKC and ERK. This is important because endothelin-1 is a secreted molecule and therefore could be used as a biomarker. And while we also discovered that endothelin antagonism is a new therapeutic strategy, either PKC or ERK antagonism actually has similar potential due to their profound suppression of endothelin-1. This also could occur potentially with fewer side effects, as bosentan is known to be fairly toxic. Thank you again for your time and attention, and for all of those who have funded this research, and I would be happy to take any questions. Please just send me an email.